my pleasure to introduce Ashley for the next talk. And I'm only there as a color commentary on this talk. You heard from Melinda about this emerging framework for exchange of clinical data. Imaging as another challenge for clinical data exchange. The reality is that the work we do is hard, and it's overlaid by the multiple efforts that all of us are engaged in, and that interface between observational research and clinical trial implementation, which is the pipeline we're all engaged in. Ashley has been an amazing thought partner in this interface between CBTN and PNOC, particularly with ATRT as being a driver use case. How do we leverage the organizational framework of PNOC as a clinical trial framework, CBTN as an observational trial framework, and cross-leverage that? She has thought more deeply and spent more time on the nuance and challenges of this space, and without solving those, I don't think we'll be successful. So it's a credit to her, and she'll educate you about that challenge.
Thank you, Adam. Thank you so much for inviting me to speak today. So Adam is going to give some color commentary at the end because he has the solutions to all of my problems. Right? Okay. So you heard a little bit yesterday about ATRT, which is a rare and deadly pediatric brain tumor. I'm going to highlight the rare because it's really important for the context of the rest of this talk. But there's only about 75 children diagnosed with this disease in the US every year. And as you heard yesterday, the majority of these occur in very young children. And we now know that there are 3 molecular subgroups. So we've taken a rare tumor and we've divided it up into even rarer subtypes. And the outcomes are poor. So despite very intensive multimodal therapy, only about 50% of these children will survive. And those that do have a lot of comorbidities. And for patients who have relapse disease, there is really no cure. So how do we approach this problem? PNOC, in collaboration with CBTN, has developed a number of disease-specific working groups. And I have the privilege of co-leading the ATRT working group with Eric Robb, who you'll hear from in a little bit. And what these working groups do is they try very hard to be as collaborative and inclusive as possible. So we meet regularly, and the we is basic scientists, physician scientists, clinicians, clinical researchers, and parent advocates. And we have a couple of our parent advocates here today. So this is what we do. We translate science. This is actually some data I think that you'll see in a little bit from Eric Robb's lab. This was work that Jeff Rubens did when he was at Hopkins. I'm looking at therapy for relapsed ATRT. And so when we have preclinical data, then the goal is to move it into clinical trials. And so at PNOC, we've developed a platform trial for patients with relapsed ATRT. It's PNOC 35. And as you can see here, there'll be a number of arms. And the first arm is going to be a combination of gemcitabine and pixelicib, again, based on the preclinical data that you see here. And the platform trial is really designed to quickly identify potential signals. So can we move something that showed promise in the lab into patients and still see some signal of efficacy? And we hope that we do. And then in that scenario, we want to move that into a clinical trial that really looks at whether or not these drugs can extend survival. So we call those time to event endpoint studies. And these are the trials that you typically hear of, like a phase 3 randomized clinical trial, which remains the gold standard. This is a problem, though, when you talk about rare tumors. So to do a randomized controlled clinical trial in a rare tumor like ATRT will take you decades. It's never been done. It never will be done. It's also questionably ethical. So what is your control arm? What are we going to give these patients? What are we going to compare them to? And is anybody willing to put their child on one of these studies? And when we talk to parents and patient advocates, the answer is no. So how do we do this better? And a number of years ago, Mike Prados and I got asked by the NBTS to help chair a research roundtable on the use of external control cohorts for pediatric brain tumor clinical trials. They actually asked Mike, and he called me and said, will you do this? And I was like, sure. And then I was like, I don't know what this is. So we started reading all this literature on it, and really found that this is a great potential use case for external control cohorts. And luckily, NBTS does a really good job of getting experts in the room that include regulatory agencies like the FDA. And so we really got to brainstorm around this with colleagues at the FDA. So just to give you a little bit more information on what this looks like. So you can see here, there's two lines. There's the orange line and the blue line. And you can imagine where we would design a trial where we would say, we want to look at agent X. And all the patients who enroll on the study are going to get agent X. And we're going to match those with patients who live in some database. And so that would be your external control. Rather than enrolling patients onto the control arm, you pull them out of a database. Sounds easy. It's not. And the reason is that the threshold for the data that need to be included in an external control cohort are incredibly high. And so this is comes from a manuscript actually written by folks at the FDA. And this is just some of the data that can be included in an external control cohort, really kind of grading those data from most desirable to least desirable. So we can hone in on the most desirable, since that's what we're going for. And the most desirable data are clinical trial data. You can imagine why. They're incredibly thorough. You can have a clearly defined patient population. You can match the eligibility criteria. And this actually was used recently in an external control cohort study that got FDA approval for DMFO for neuroblastoma. So they used clinical trial data as their external control cohort. They were very fortunate to have those data. So what about for ATRT? Well, there's only been really 3 prospective studies for ATRT, only really 1 study, maybe 2 for relapsed disease. And we don't have the patient level data for those studies, and they're outdated. So that as a data source for us, not great. And then you could say, okay, well, what about a new ATRT study? Well, there isn't one. So COG had one planned for upfront therapy. The drug company is no longer supportive of that clinical trial. So realistically, there isn't going to be another study for upfront ATRT. I think a decade is a reasonable amount of time before we see that happen. Actually, that would be nice. But I'm doubtful that that will happen. And then as far as clinical trials for relapse ATRT, there really aren't any right now. There's a PEPN study through COG, and that's really about it. So we don't have those data. So then we move on to kind of our next best, which is prospective cohort or registry data. And fortunately, we do have patient level data at CBTN, and we have the ability to get more. So this is where we really focused. So what does that look like? So we developed this ATRT registry in close collaboration with CBTN. It's PNOC 30, and I'll kind of walk you through it. Let's see, I have a cursor. So the eligibility is pretty broad. You have to have a diagnosis of ATRT. Patients who were previously treated, patients who were deceased, their parents can enter their data into the registry. Patients with newly diagnosed disease or relapsed disease are also eligible. But you need to have tissue available and or molecular subgroup information available by some means. Patients can enroll through a number of ways, and I'll show you that on another slide. But basically, you can have patients from the CBTN cohort that exists, and then they can enroll directly onto the registry through PNOC or through a central enrollment site through Children's Hospital Los Angeles. Once patients are enrolled, we collect a lot of data that's on the left. So there's clinical data, there's imaging, which we will have reviewed centrally, which I think is a really important point here. We'll collect plasma when available, CSF for prospective patients at time points that match imaging when possible. We know that CSF liquid biopsy is something that we need to continue to investigate. Again, if we don't have molecular classification, we'll collect tissue and have that done. There are quality of life assessments and social determinants of health that will be gathered as well. If we move to the middle of the slide, patients who are still undergoing treatment or finish their treatment will continue to be followed in a prospective manner. And if at any point a patient has relapsed disease, we will offer them an optional expert tumor board. And again, this really came out of conversations with our advocates who said, we're asking for multiple second opinions from you guys around the country. This is really hard. And we said, okay, well, we all sit together and talk about this registry. We'll just review the information all together with an expert neuroradiologist, and then we'll give an opinion back to your treating physician to review. So again, just trying to really incorporate what we're hearing from our advocates into our design. And then really important is the data sharing, right? So you've been hearing this over and over, and our goal is to share these data with anybody who needs them. So these will go through typical CBTN sharing mechanisms that you see here. Okay, so sounds really good. At least I think it sounds really good, and we're really excited. It's just not that easy. So one of the biggest challenges is the differences in data requirements. And something that we talk about in CBTN and our working groups is data tiers. So how much data do you need to do project X? How much data do you need to do project Y? Do you need just the molecular? Do you need molecular and survival? And what about for an external control cohort? So this is I pulled this is the chemotherapy CRF from CBTN, and it's really detailed. These are the data elements that we need for the external control cohort, and this came out of conversations with the FDA. So the problem is there's a delta, and the delta is not that small. So CBTN collects so much data, yet the threshold for this external control cohort is incredibly high. So we run into this as an issue, and we have this amazing, CBTN cohort of patients, but we really have to get this additional information in order to make this useful in this context. The other issue is enrollment. So how do patients enter? So if a patient is being treated at a site that's only a PNOC site and isn't a CBTN site, it's pretty easy. They enroll in PNOC 30. If they're both, it's a little bit more complicated. Do they enroll through CBTN? Do they enroll through PNOC 30? Who's doing the data collection? Then who's sharing it with who? They're a little bit different platforms. How are we going to get those data to crosstalk? And then if you are a CBTN site only, then do you enroll just in CBTN? Do you enroll just in PNOC, or do you enroll in CBTN and PNOC 30? And if you're enrolled in both, again, who's doing the data collection and how are we sharing those back and forth? And what about the patient who's treated at multiple institutions? A lot of these patients are treated at multiple institutions. They start somewhere, they move, they go get a second opinion, and we're still kind of reconciling that issue. Then there's the data sharing across platforms. So certain platforms allow collection of PHI. Then do we share PHI? Then what if the patient had MCI done and we're trying to cross-link all these patients' data across multiple platforms? And then what agreements exist and what don't? And you can say, oh, well, just get a two-way agreement. And well, it's a little bit more challenging than you might expect. So I put the star up because this seems to be the easiest agreement out of all of these, yet is actually quite challenging. So some other challenges. I think you've heard this before, and I so appreciated the speaker yesterday who pointed out all of the hours that people spend importing data into registries. And I died a little bit in my seat because this is really an issue. And this is really the resources, right? This is where the money and the time are going. And this is, as you may be able to tell, a big source of frustration. So let's just take a few examples. If we have a patient who's treated at a PNOC institution, there's a PNOC coordinator who is going to go through that patient's medical record, and they are going to upload all of these data elements into the database. What if the patient wasn't treated at a PNOC institution? The owner is really on the family. So the family has the burden at this point to go and collect all this medical information, give it to a coordinator who's sitting at CHLA. That coordinator is going to comb through all of this information. I will oversee all of this being combed over and then inputted into this clinical trials database. And then you've got these other patients, right, who are CBTN, and we talked about that. So even if it's a CBTN site that's not a PNOC site, you have a CBTN coordinator that's putting in all this information, and we're looking at what the delta is and then trying to figure out, how to complete this record. Then PNOC has to do, we have to QC the data, right? So do we actually have all the imaging? Maybe, maybe not. Do we have all the clinical data? Maybe, maybe not. And do all those data make sense in the way that whoever manually uploaded them? Does the story make sense? Oops, missed my point, which is this is really the time suck and the money suck. It's really overwhelming, the amount of time and money that this takes. And this is about ATRT, but it's not. So we didn't go on this massive journey just for ATRT. It's a worthy cause, but there are a lot of worthy causes, and there are a lot of these rare diseases that need similar efforts. So if we start with ATRT, then we move on to choroid plexus carcinoma, pineoblastoma, ETMR. We've got subgroups of medulla. We're having the same issues. And there is kind of an ability to generalize some of this, but not all of it. So there are going to be specific data elements that you need for ATRT that are different than what you need for ETMR. That are different than what you need for CPC. And this is really where the clinical researchers come in, and their input is really, really important. And then I mentioned alignment. So we need to make sure that we're kind of all moving together. There's really, I intentionally made this kind of like a wheel, because we don't need to keep reinventing the wheel, or we need to make the wheel better, but we need to take what we've got and really augment it rather than kind of doing this over and over and over again. Before we take a break, Adam is going to fix this.
So when Ashley and I discussed, she said, you have to talk with me because I'm not sure I can explain it all, but you explained it all. I think this is one of the, key opportunities for us. As Ashley mentioned, it's not just for ATRT. This is going to be an existential challenge across the clinical trial landscape, and there are several layers in there. I just want to connect this to some of what you heard yesterday and what you'll hear today. So yesterday we heard, on the ARPA H session, right, these efforts by No2 and MuleSoft, these workflows of connecting data between institutions, of investigating the potential of leveraging TEFCA-based architecture with No2, like Melinda just mentioned, to address how do you bring in data from institutions that the patients were in. Today, you'll hear from Tamara on extracting data more natively from the EHR to support this. So again, I think that's the first pillar challenge of just how do we get all the data in ways that can allow us to choose the elements that are relevant for ATRT, choose the elements that might be relevant for craniopharyngioma, right? But have a common framework for that process. Because the human-based architecture, even for 75 patients a year, is not sustainable, right, as a framework longer term. The second part that you're hearing is that regulatory governance, management of patient IDs and PHI transfer and connectivity around that context. You heard a tiny bit yesterday about the work we're doing with NCI, and Sarah talked about MCI, the Medical Characterization Initiative, and how we're going to try and connect IDs between MCI, CBTN, PNOC as a context. these are just touch points of what you've heard over the last day and today about the efforts that I'm not going to fix, but we're going to fix, right, as a representational context for all opportunities in this setting. ATRT will be a use case in our ARPA-H initiatives, right? We'll test, vet, share along this map that Ashley just shared, right? What problems can we solve? What are not solvable? What do we have to come to Melinda with and say, can you help us fix this in order to advance this process? I think it's such an amazing use case, and you've done amazing work trying to galvanize it. I think it's going to help everybody. So thank you so much.